The story of James Watson and Francis Crick, the double helix that changed biology forever. In the early 1950s, biology was at a crossroads. Scientists knew that genes carried the instructions for life, but no one could figure out exactly how that information was stored and passed on from one generation to the next. Everyone agreed the answer lay inside DNA, deoxyribonucleic acid, a molecule that had been discovered decades earlier but remained a mystery. The race begins. James Watson, a young American biologist with a sharp mind and a restless energy, arrived at the University of Cambridge in England in 1951. There, he met Francis Crick, an older, exuberant British physicist-turned-biologist who loved big ideas and loud debates. At the time, several research teams around the world, including one led by Linus Pauling in the USA and another at King's College London, were competing to unlock the secret of DNA's structure. Whoever succeeded would revolutionize science. Watson and Crick believed that if they could figure out the shape of DNA, they could explain how it worked. But they didn't have the best experimental data. Instead, they had brains, boldness, and the ability to piece together clues like detectives in a molecular mystery. A crucial clue from X-ray images. At King's College London, Rosalind Franklin and Maurice Wilkins were using a cutting-edge technique called X-ray crystallography to take photographs of DNA fibers. One of Franklin's images, later known as Photo 51, revealed a striking pattern, an X shape that hinted at a helical structure. In early 1953, Watson saw the image without Franklin's direct permission, a controversial moment in scientific history. That photograph was the missing piece they needed. Building the model. Watson and Crick rushed to build a physical model of DNA using metal rods and plates to represent atoms and bonds. They worked for weeks, arranging and rearranging the pieces. Finally, they saw it, a double helix like a twisted ladder. The two rails of the ladder were made of sugar phosphate backbones. The rungs were pairs of nitrogenous bases, adenine with thymine and cytosine with guanine, always in these pairs. This pairing explained how DNA could replicate. Each strand served as a template for making a new one. The announcement. On April 25, 1953, Watson and Crick's paper was published in Nature. It began humbly. We wish to suggest a structure for the salt of deoxyribonucleic acid DNA. This structure has novel features which are of considerable biological interest. Behind those calm words was a discovery that transformed biology, medicine, and genetics forever. Legacy and controversy. In 1962, Watson, Crick, and Wilkins